Hereby, I open this academic ceremony in which Mr. Matthijs de Rijk will defend his academic thesis entitled Interactive Control of the Lower Urinary Tract, Translational Models in Functional and Neurology. May I invite you, Mr. de Rijk, to present a summary of your study and the conclusions of your thesis in the next 15 minutes. Thank you very much.
Dank je. Beste prorector, beste leden van de Corona, vrienden, familie en collega's. In de komende 15 minuten wil ik jullie kort wat meer vertellen over het onderzoek dat heeft geleid op mijn proefschrift met de titel Interactieve controle over de lage urinewegen, translationele modellen in functionele en neurourologie. We zijn allemaal bekend met het gevoel om heel nodig naar de wc te moeten en nog meer iets bezig te zijn. En dat plassen nog even uit te stellen totdat je af hebt gerond waar je mee bezig bent. En voor de meeste van ons is dat iets heel vanzelfsprekends. Maar stiekem zijn er een hele hoop ingewikkelde processen die daaraan ten grondslag liggen. En die op de achtergrond ervoor zorgen dat we dat ook daadwerkelijk kunnen. En voor veel mensen is dit ook niet vanzelfsprekend. Om die patiënten beter te kunnen helpen, is het belangrijk dat we wat meer te weten komen over de precieze controle van de lage urinewegen, de processen die daaraan achter schuil gaan. En dat is wat ik heb geprobeerd in mijn onderzoek, om daar een beter beeld over te krijgen. Want de lage urinewegen zijn op te delen in 2 functionele gebieden. Dat is de blaas zelf, het gebied dat verantwoordelijk is voor de opslag van urine. En de sluitspier en de plasbuis. Dat is het gebied wat verantwoordelijk is om de blaas gesloten te houden tijdens de vulfase, tijdens het vullen van de blaas. De wand van de blaas die is op te delen in verschillende lagen. Dit plaatje is een kwartslag gedraaid, dus de bovenkant is de binnenkant van de blaas en de onderkant is de buitenkant van de blaas. En de binnenste laag, dat is het uroteel. En deze laag heeft een signalerings- en barrièrefunctie. En de signaleringsfunctie, dat wil zeggen dat deze laag verantwoordelijk is om via chemische interactie informatie door te geven naar andere lagen. En barrièrefunctie, dat betekent dat deze laag er verantwoordelijk is om ervoor te zorgen dat urine niet het onderliggende weefsel binnen kan dringen. De volgende laag, dat heet de lamina propria. En deze laag bestaat uit veel elastische vezels. Die is verantwoordelijk voor de rekbaarheid van de blaas. En hier zitten veel zenuwvezels in. Dan de volgende laag is de detrusor. Dit is de daadwerkelijke blaasspier. En die bestaat uit spierlagen die in verschillende richtingen lopen. En dit gebied is verantwoordelijk om de blaas als het ware leeg te knijpen. De lage urinewegen hebben 2 verschillende functies, namelijk de opslag en de uitscheiding van urine. En het uitscheiden dient alleen maar te gebeuren wanneer de blaas voldoende gevuld is en wanneer we vrijwillig het plassen willen starten. Nou, tijdens de opslag heeft de detrusor is dan ontspannen om zo een groot reservoir te vormen. En het gebied van de plasbuis en de sluitspier is dan aangespannen om te voorkomen dat urine uit de blaas lekt. Tijdens de uitscheiding is dat precies tegenovergesteld. Dan ontspant de sluitspier en opent de uretra. En dan zal de blaasspier, de detrusor, aanspannen om de blaas leeg te knijpen. Nou, om ervoor te zorgen dat dit correct gebeurt, is er een continue interactie tussen het centrale zenuwstelsel, de hersenen en de blaas. En dus een continue uitwisseling van informatie. En het optimaal functioneren van dit systeem is belangrijk voor een correcte uitvoering van die blaascontrole. Dus van het opslaan, van het uitscheiden van urine. En afwijkingen in de functie of in de structuur op een van deze niveaus, of op de interactie tussen deze niveaus, kan leiden tot problematiek bij opslag of uitscheiding van urine, plasproblematiek. En deze dysfunctie komt heel veel voor in de samenleving. Het neemt enorm toe met veroudering. En omdat we allemaal, onze levensverwachting wordt hoger, dus ook de prevalentie hiervan neemt enorm toe. En het heeft een hele hoge impact op de kwaliteit van leven van patiënten en de zelfredzaamheid van patiënten. Het onderzoek dat ik heb uitgevoerd centreert zich rond 2 heel brede onderzoeksvragen. En de eerste vraag is, wat is de relatie tussen plasproblemen en de structuur en de functie van de blaaswand en de bekkenbodem? Dat focust zich dus op lokaal niveau. En de tweede vraag is, kunnen we blaasgevoel meten in de hersenstam? Dat focust zich dus op centraal niveau. Nou, het proefschrift bestaat uit 5 verschillende studies, die ieder terug te leiden zijn naar een van deze 2 brede onderzoeksvragen. Als eerste hebben we gekeken naar de effecten van veroudering op de structuur en de functie van urotheelcellen. Dus dat is op het lokaal niveau in de blaas. Vervolgens hebben we gekeken naar de effecten van langdurige psychologische stress op de gezondheid van de blaashals. Dat is een subgebied van de blaas zelf. Dan hebben we gekeken naar werkingsmechanismen die onderliggend zijn aan het effect van een therapie voor stressincontinentie. En daarbij focussen we met name op de ondersteunende structuren van de blaas in de bekkenbodem. Vervolgens hebben we gekeken naar een functionele opdeling van hersenstamstructuren die betrokken zijn bij de controle van de blaas en bij blaasgevoel. En als laatste hebben we gekeken naar de consistentie van die functionele opdeling op groepsniveau. En voor ieder van de 2 brede onderzoeksvragen zal ik een studie die daaraan gerelateerd is wat verder belichten. Als eerste presenteer ik een studie die te koppelen is aan de eerste onderzoeksvraag en zich dus focust op het lokaal niveau. We kijken hierbij specifiek naar het urotheel. En dat is dus de laag die als functie die signalerings- en barrièrefunctie heeft. We hebben hiervoor gebruik gemaakt van urotheelcellen, urotheelweefsel verzameld van jonge en oude ratten. En we zien in dit weefsel een afname van de gezondheid van cellen. Daarnaast zien we een afname van de gezondheid van de mitochondriën. Dat zijn de energiefabriekjes voor cellen. En op dit plaatje, waarin we gezonde mitochondriën hebben aangekleurd, zien we een veel sterkere reactie in het linkerplaatje. Dat is van jonge dieren, dan in het rechterplaatje. Je ziet dat dat veel donkerder is. En dat betekent dat de mitochondriën in dat weefsel minder gezond zijn. Daarmee samenhangend hebben we ook gezien dat er een afname is van het vrijkomen van ATP. En ATP is de drager van chemische energie voor de cel. Nou, samengevat kunnen we hieruit afleiden dat veroudering negatieve veranderingen in het urotheel teweeg brengt. En dat kan de capaciteiten van het urotheel om die signalerings- en die barrièrefuncties optimaal uit te voeren verminderen. En dit heeft zodoende dus ook een impact op de gezondheid en het functioneren van de lage urinewegen. Ik zal nu wat verder ingaan op een studie die te linken is aan de tweede onderzoeksvraag, op het centraal niveau. En een gebiedje in de hersenen, een bijzonder belangrijk gebied in de hersenen voor blaascontrole is het periaqueductaal grijs. Afgekort tot PRG. En dat zit verborgen diep in de hersenen. Ik heb dat hier met rood gemarkeerd op een MRI-scan van een van onze proefpersonen. Als we verder inzoomen op het PRG, dan zie je echt dat dat heel donker is ten opzichte van het omliggende weefsel. Het PRG zit ongeveer op dit niveau in de hersenstam, die groene vlek op het plaatje aan de rechterkant. En het ontvangt informatie van de blaas over hoe vol de blaas is en het stuurt dat vervolgens rond naar hogere hersengebieden die een beslissing maken over wanneer we naar de wc moeten gaan. En vervolgens stuurt het PRG deze informatie ook weer terug richting de blaas. Het PRG functioneert dus als een soort van schakelstation. En op deze manier is het PRG een beetje te vergelijken met het Utrecht Centraal Station. Als je door Nederland reist van noord naar zuid of van zuid naar noord, dan is Utrecht Centraal een station waar je heel vaak over moet stappen. En dat is hetzelfde voor informatie die van de blaas naar de hogere hersengebieden reist. En in ons onderzoek proberen we het PRG een beetje op dezelfde manier te benaderen. En op het moment dat je weet hoe informatie zich verplaatst door dit schakelstation, dan weet je een beetje wat er gebeurt in het grotere systeem, over het hele spoorwegnet. Hiertoe hebben we vrijwilligers gevraagd om deel te nemen aan een fMRI-studie, een functionele MRI-studie. En middels fMRI kunnen we hersenactiviteit meten. Een fMRI-scan maakt driedimensionale beelden waaruit hersenactiviteit af te leiden is. En deze beelden bestaan uit voxels, dat zijn een soort van 3D-pixels, waarin informatie over hersenactiviteit zit. En we verzamelen deze informatie voor een heleboel verschillende voxels. En zo hebben we informatie over hersenactiviteit in een groter gebied van de hersenen. En deze informatie verzamelen we gelijktijdig met het vullen van de blaas via een katheter en met het meten van blaassensaties door vragenlijsten die de proefpersoon bijhoudt met een joystick. Vervolgens hebben we voor alle voxels uit het PRG gekeken hoeveel de activiteit in die voxel lijkt op de activiteit in andere voxels. En op basis daarvan hebben we een aantal groepen, clusters kunnen maken van voxels die een vergelijkbaar patroon van hersenactiviteit laten zien. En die voxels hebben we hier dezelfde kleur gegeven. En we verwachten dat deze opdeling van het PRG gekoppeld is aan de anatomische opdeling van dit gebied. En om dit verder te toetsen hebben we een aantal analyses gedaan. Zo hebben we gekeken naar de symmetrie van dit gebied, omdat we weten vanuit proefdieronderzoek dat het PRG symmetrisch opgedeeld is. En in onze data zien we inderdaad ook dat er meer symmetrie is dan je zou verwachten op basis van kans. Vervolgens hebben we ook gekeken of de opdeling die we maken op een lege blaas en op een volle blaas, op fMRI-data, of die op elkaar lijken. En dat is wat je zou verwachten, omdat de onderliggende anatomie immers gelijk blijft. En we zien inderdaad ook dat deze opdelingen heel veel op elkaar lijken, meer dan je zou verwachten op basis van kans. Vervolgens hebben we ook gekeken hoe de communicatie tussen die verschillende clusters verandert als een functie van verandering in blaasgevoel. En dat hebben we gedaan met de data die we hebben verzameld middels de joystick. We hebben dit dan opgedeeld in verschillende sensorische staten en we zien inderdaad dat voor verschillende clusters de onderlinge communicatie verandert tijdens veranderingen in blaasgevoel. Wanneer we de blaas dan beschouwen als station Maastricht, het PRG is station Utrecht Centraal, het belangrijke schakelstation, en de hogere rechtsgebieden als Den Haag, waar de beslissingen genomen worden, dan kan de methode die we hebben ontwikkeld ons helpen om in kaart te brengen hoe passagiers vanuit Maastricht door het station Utrecht Centraal, het PRG, bewegen om de trein naar Den Haag, naar de hogere hersengebieden te nemen. De ontwikkelingen die we hebben gedaan geven veel mogelijkheden om controle over de lage urinewegen beter te onderzoeken en beter te leren begrijpen. En we zullen in de komende jaren ook, mede dankzij een toegekende beurs, nog verder gaan met vervolgonderzoek. Zo zullen we gaan kijken naar hoe de communicatie tussen het PRG en die verschillende clusters is met andere hersengebieden. We zullen gaan kijken of er verschillen zijn te vinden tussen gezonde mensen en patiënten met dysfunctie van de lage urinewegen. En we zullen gaan kijken naar wat de effecten zijn van succesvolle, maar ook onsuccesvolle behandelingen op de communicatie tussen hersengebieden. Nou, de belangrijkste conclusies die volgen uit dit onderzoek zijn dat plasproblemen gepaard gaan met negatieve veranderingen in de blaaswand, die mogelijk verbonden zijn met veranderingen op het niveau van het centrale zenuwstelsel. En het functioneel opdelen van het PRG op basis van fMRI-data biedt nieuwe mogelijkheden om de controle van de lage urinewegen beter te leren begrijpen. Bedankt voor jullie aandacht en ik geef het woord graag terug aan de prorector.
Dank u wel, meneer de kandidaat. Mooi in tijd. En voor de duidelijkheid, we switch now to English.
Yeah.
Yes. So I also would like to mention to our audience that the thesis defense this afternoon will be complete in English because we also have some guests both from the UK as well as from the United States available online, which will be on the screen on my right side and later on the screen here in the front as well. We have 7 members in the Corona, so we have about quite a lot of questions who can be addressed and who can be answered by you. And the opposition then will be opened by Professor Linden. Professor Linden was the chairman of your assessment committee, and he's affiliated as scientific director of the School of Mens. And I would like to give him the first opportunity to discuss with you.
Thank you. Dear candidate, as someone who's worked on functional MRI for more than 25 years, but never on bladder control or topics related to urology, I would like to congratulate you and your promotion team that you've taken up the challenge of studying this very important, clinically and physiologically important system, which is obviously understudied, and perhaps some of the challenges you encountered in your thesis and your work explain why so few groups take up this challenge. But you have, and that's great. And I have questions about the fMRI work, in particular, chapter 6 in particular. So in a conventional fMRI experiment, you would use a block design where you compare on and off periods. Now, that is probably not possible or not straightforward in bladder protocols and bladder filling protocols, so you chose another approach. Could you perhaps explain what the challenge is? I mean, you can't just switch back and forth from filling state and empty state, but what are the challenges, and how could you approach this kind of challenge of getting more into comparison between a full and empty state?
Yeah. Highly esteemed opponent, thank you for your compliments, and thank you very much for this really interesting question. This is something we also asked ourselves very, very early on, of course. And in fMRI research in the field of urology, some block designs are used. Some groups infuse and withdraw saline into the bladder repetitively, very quickly, but this induces sensory artifacts. And that's something we really wanted to try to avoid. So we're trying to mimic a natural situation as closely as possible, while still having some control over the volume in the bladder. So that was our main reason for the approach that we took. Yeah, and so we decided to continue with these resting state scans, and this is well, it's kind of a groundwork to try to start to understand the functional organization of the PAG and try to map changes in communication between these clusters. But I think block designs are sooner or later necessary, and I think a very interesting block design would be to repetitively have participants void and interrupt their voiding for a number of times, and combine that with resting state approaches.
Thank you. Very clear. And now you had this nice analogy of the Dutch railway system. Now, Utrecht Central, of course, is a study that's very well known to everyone who's ever tried to get from A to B in the Netherlands. So I appreciate the interest in Utrecht Central and its track system. But of course, ultimately, we may want to get to The Hague or Amsterdam. So could you speculate, or could you sort of perhaps sort of elaborate on the further connectivity? I mean, you had limitations from the particular scanning protocol, but are you also planning to move towards the sort of cortical areas that the PAG connects to, or other brain areas?
Yeah, we are definitely planning to move to other brain areas, but there's also, in my analogy with Utrecht Centraal, I think that if you know how information or how people move through this central relay station, then that kind of reflects what's happening in the larger network. So based on that information within Utrecht Centraal, you can tell what train has recently arrived and what train that information or those people are trying to switch to. So I think that looking at the PAG will give us more information about the whole system as well. But we're very much, the first step that we're going to take is looking at interaction between the PAG and the ponds, where the pontine mixturation and storage centers are located. And we're going to, in the continuation of this project, we're also going to try to integrate our findings with MRI scanning at the level of the whole brain.
And then perhaps my last question, if you want to look at the connectivity further on, will this be based on functional connectivity, or is there a role for diffusion spectrography, or rather the pathway is not specific enough, white matter connections?
I think we will continue with functional connectivity. Yeah, that's an approach that we've been taking, and I think that seems to reflect. We have some pilot data also looking at the interaction between the PAG and the ponds, and we're getting more and more enthusiastic about how meaningful this functional subdivision of the PAG is, and I think it's a really fruitful approach that we've taken.
Perhaps a very final, very quick final question, because you were so quick in answering. What if you had one cortical area that you could sort of measure in connection with these sort of brainstem areas? Which one would that be?
I would first, of course, look at the insula and the anterior cingulate cortex, which are the sensory and motor areas for the autonomic nervous system. I think that would be very interesting areas to look at, and of course, the thalamus as a subcortical structure should be included as well.
Thank you very much. And great answers. I hand the word back to the pro-rector.
Thank you very much. The opposition will be continued by Professor Penniker. He is affiliated with the Department of the National Hospital for Neurology and Neurosurgery of the University College of London. And of course, I would like to thank him that he joins this afternoon. Professor Penniker, I'd like to give you the word.
Well done, thesis. I have a couple of questions here. One is in regards to sort of potential bias. When we think about the PAG, we know it's involved in interoception, which is what your thesis is about, but we also know about nociception and also defensive behavior in terms of sort of anxiety and other emotions. Now, here we are with an experiment that's run in a very hostile environment, hostile to the healthy control in regards to an MRI scanner, that can bring on fear, anxiety, the noise, claustrophobia, but also internal threats as the bladder is getting filled and it reaches that strong desire of urgency, of course, the emotions associated with that. So in this study, I was wondering, how did you actually cluster, how did you actually control for these additional threats when we look at sort of changes in neuronal activity in the resting state and in regards to just bladder filling alone, but there are numerous other threats that could also be resulting in activation in these different clusters.
Highly esteemed opponent, thank you very much for this question. This is also a very relevant and interesting topic. For looking solely at the PAG, we've not been very concerned about this. We've moved to including measurements of respiration and heart rate to filter some of these autonomic processes out of our data. But now that we're starting to look at the interaction between the PAG and the PONS, for example, and we see in our pilot data, we see multiple areas in the PONS that show changes in functional connectivity with different clusters of the PAG. And it may very well be indeed that these different areas that we see in the PONS are involved with different types of autonomic functions. And now it does indeed become very relevant to try to distinguish which autonomic process is represented where in the PONS. Because, yeah, like you mentioned, it may indeed be that along with an increase in desire to void and bladder fullness, participants will report an increase, or will show an increase in heart rate. They will get a little bit sweaty. They will get a little bit nervous, indeed, worried that they will accidentally void in the scanner. And yeah, those are indeed challenges that we're encountering now.
So say, for example, your next project is related to another sort of stimulus, for example, what pain research, or perhaps maybe on bowel interoception. And if you were to have a similar sort of a paradigm, but of course with a different way of stimulation, would you expect to find similar clusters of activation? So in this case, it's specifically bladder filling. But what might you expect? What can you speculate might happen if there was another type of stimulation?
Within the PAG, you mean?
Within the PAG, indeed, yes.
Within the PAG, I would expect that our subdivision just represents a functional subdivision of the PAG. So regardless of stimulus, I expect that the subdivision will remain the same. But interaction between clusters may look different indeed for different types of stimuli. And I think pain may indeed be very relevant and interesting to study in this respect. And the interaction, of course, between the different clusters of the PAG and other brain areas will differ. We expect that, we know that the PAG is organized in sympathetic and parasympathetic areas, columns, ventrolateral and dorsolateral column. And we think that the functional clusters that we were able to delineate from our fMRI data can represent sympathetic and parasympathetic areas. So with that respect, you would kind of expect sympathetic functions to always be represented in this in a specific functional cluster that we were able to delineate, and vice versa for parasympathetic functions.
Thank you. And my second question was regarding the clustering and the connectivity. So in I believe it's chapter 6, you demonstrated the greatest correlation between clusters 2 and 3. But that correlation was greatest with the first sensation. And then when it went to first desire and strong desire, actually, there was no correlation at all. And what are your thoughts about this, and what is the relevance of that finding, do you think?
I think first and foremost, the finding that we illustrate in this figure should be taken as a proof of concept. So I think we need to repeat this in many more participants, and we need to optimize the experimental protocol in order to do so. But I do think it's very interesting that this correlation seems to be highest at the first sensation of bladder filling. But in order to draw strong conclusions from that, we really do need to include a lot more participants in a study like that.
Great. Well, thank you. Thank you very much.
Thank
you. Then the opposition will be continued by Professor Joosten. He's affiliated with the Department of Anesthesiology at our university, and I'd like to give him the word.
Thank you. I would like to congratulate the team with its excellent work. And I would like to discuss with you about the PAG. The PAG, and you made an analogy with Utrecht. When I travel from Maastricht to The Hague, I never go via Utrecht. I always go via Breda or Rotterdam. So, and on the other hand, if you then place the bladder in Maastricht and the higher cortical levels in Holland, next time you could change it and place the bladder in Holland and the higher cortical levels in Limburg. That would help me. Anyway, the PAG is a very, very complex network. It's also involved in pain. That's my field. Areas like the dorsolateral and the ventrolateral parts of the PAG are known to be involved in pain, in chronic pain, and they are also stimulated with electrodes to help people who do have pain. And then I come to your, you have this fMRI, which in fact only tells there is more blood and not really something about functionality, whether there is inhibitory or excitatory interactions and connections with other areas. And so what does help this, let's say, this approach? You show this clustering. We do know from, let's say, the old neuroanatomy how and what is in the PAG, serotonergic connections with other brain areas. And so could you superimpose your findings to the, let's say, the neuroanatomy as we know from the PAG, and what in that context will be the future? Because you say, yeah, I want to study connections with the pons, connections with the thalamus, which is rather obvious. These are all kind of Utrecht central stations, by the way.
Thank you very much, highly esteemed opponent, for your very interesting question. I think it is, so to some extent, resting state-based parcellations are known to reflect the underlying anatomical organization. And it's very difficult, so like I also tried to mention in my thesis, it's difficult to investigate how our results relate to post-mortem anatomical studies of the PAG with staining of different types of neurons that express different transmitters, because we don't know how they interact in the living state. And it's also very evident that we don't see in our clustering, we don't see the very common columnar organization of the PAG. And I argue that is because we look at the PAG in a completely different state, and there are studies that also show that there is interaction across columns with regards to information, and also along the craniocaudal axis there are subclusters within the PAG that are functionally unique within their own column, and I think that we study the PAG in an active state where all of these processes are ongoing, and we are able to parcellate, functionally subdivide the PAG based on this resting state data.
Would it not help to let's say to combine, let's say, the knowledge we have from the PAG, this complex area, and the projections, for instance, serotonergic, I refer to that serotonergic projections to the RVM, which we know are important in the pain, but obviously or probably there will also be certain known projections, known neurotransmitters, known inhibitory or excitatory neurotransmitter relations with other areas, and use that in addition to this, and then maybe you end up with these parcellations, but maybe not, or you stick with these parcels.
I do think it's a very good idea to try to integrate two with each other. So.
Well, sorry, you said not a good idea, or was it?
I do think it's a good idea.
Thank you.
Yeah, but but of course that's very difficult in the participants that we include in our studies. So you would have to switch to animal models or gather post-mortem human tissue. You could not do that within the same participant. But another thing is that for these different the anatomically known columns, we know that there's different interaction patterns with the rest of the brain for distinct columns, and that is something we can look at with functional MRI as well. So we can, if we think that one of these clusters represents a parasympathetic area of the PAG, which may correspond to the ventrolateral column, then we can look if the interaction pattern of that cluster with those voxels that we see shows a similar interaction with the rest of the brain that we know from anatomical work. So I think both of these approaches are helpful to gain a complete understanding of the PAG.
I have still sometimes it's interesting to see that then projections, and you mentioned that also to the anterior cingulate cortex, the insula, also in the pain. From the PAG, they project to the same areas. So that really confuses me and say, okay, is this PAG where so much comes in and out, and it's related to pain. You refer somewhere to Alzheimer's disease, reference number 30 on page 76, also in the PAG. So there is so much in these areas that is being relayed and integrated that it will be extremely difficult to focus on this subcortical area. So my question is, there is so much, and do you really think that if stimulating these areas, these parcels in the future, if you want to stimulate that you will have a lot of side effects and a lot of complications, which finally will end up in a no-go?
Yeah, I think you would, and I would not. Stimulating the PAG would not be my first thought. Okay. I would more try to use the PAG as a location to measure therapeutic effects, and try to optimize your therapies based on what you see in the PAG. But I would not stimulating the PAG directly would not be my first thought.
Okay, thank you. I the word back to the prorector.
Thank you very much. Then the opposition will be continued by Dr. Blok. He is affiliated with as urologist at the Erasmus Medical Center in Rotterdam. And of course, I also like to thank him that he joins us today all the way from Rotterdam. Thank you very much.
Thank you very much. It's my pleasure to be here. I also congratulate you with your fine work. I have a question about the chapter on the balloon, chapter 4. I was intrigued by the working mechanism. How can you treat with an air-filled balloon stress urinary incontinence? Is it elongation or obstruction or something else? Could you elaborate about that?
Esteemed opponent, thank you very much for this question. Yeah, we were also very much intrigued by the working mechanisms behind the balloon. So the company that designed this balloon proposed that the balloon, because gas compresses more easily than liquids, so the gas, the air inside the balloon would compress before the liquid in the bladder would compress. So in that sense, it would act as a sort of a shock absorber within the bladder, and that would decrease the difference between the increase in abdominal pressure and the increase in intravesical pressure during an episode of high abdominal pressure during sneezing, coughing, standing up. And that was their proposed working mechanism. But it's very difficult to assess this in humans, because you would have to check or measure intravesical pressures and intra-abdominal pressures before and after implantation of this balloon. But it's impossible to have someone cough twice with exactly the same magnitude and the same pressures. So it was difficult to directly assess that proposed mechanism. So what we decided to do instead is to assess the surface area of the balloon in the video aerodynamic data that we collected, and we did see a significant decrease of the area of the balloon. So that does show that to some extent it absorbs some of the increase in abdominal pressure. But what we thought was very interesting is that we also saw changes in the shape of the bladder, and we saw changes in the movement of the area of the bladder neck. And we tried to quantify those changes and link those to proposed working mechanisms. And what we propose is that the implantation of the balloon changes the shape of the bladder, and that improves it increases the mobility of the bladder neck, and that may help to make the kinking of the urethra. So the point in the urethra within the urethra, the point that reaches the highest pressure during an episode of high abdominal pressure is more easily exposed to these increases in abdominal pressure, and it reaches that point more easily. That's what we think may contribute to this effect.
Okay, thank you. And another question about this chapter is, what do you think is the position in the care pathway of this balloon? Is it very in first line treatment or third line? I'm a little bit confused because it is an invasive therapy, while you call it an implantation. Maybe it's an insertion of a balloon, not an implantation.
Yeah.
Could you help me with that?
Yeah, that's very, very difficult for me as a basic scientist to really determine. But I do know that some patients tolerated this balloon quite well, and for some it was a useful therapy for a period of time. But I think almost all of them had a balloon removed after some time, and the company that developed this therapy also in the end did not get FDA approval for the device. But I do think that the studies that we were able to do, the mechanism that they propose does have a therapeutic effect, and I think understanding or the therapy that they develop does have an effect, and if we can understand the mechanisms that underlie that effect, that will really help us to understand stress incontinence better. And even if this was not a very at least at this point, is not a very commonly used and successful therapy. It will help us to improve the therapeutic options for stress incontinence.
Okay, thank you very much. Am I allowed?
There is room for one more question.
Yes. I was also very intrigued, maybe as you know, about the discrepancy about columnar organization, post-mortem, animal experiments, and your own results. And could you help me again? I see a little bit a discrepancy between chapter 5 and 6. That in chapter 5, it's on the PHE, you see some kind of columnar organization, and chapter 6, if you look at the pictures, is a little bit different. Could you help me with that? Because I see absence of columnar organization in the last chapter, while you have some in the fifth chapter.
Thank you very much for your question. I do think it's difficult to say whether in either of these chapters you see a columnar organization. I don't think it's not being quantified, and I think visually it's very difficult to say something about a three-dimensional structure just in a 2D picture. What we do see, and that's not a discrepancy between chapter 5 and 6 at all, because in chapter 6 we show that for many of these clusters we find a large enough area of overlap that is significant across participants. So I think that actually shows that there is a very high agreement between chapter 5 and chapter 6.
Okay, thank you very much.
Thank you very much. Then the opposition will be continued by Dr. Stiers. He is affiliated with the Department of Neuropsychology at our university, and I also like to give him the word.
Dear candidate, I first want to congratulate you with a very fine imaging study that you did. I also have an imaging background, so I will also ask questions about chapter 5 and 6, of course. That is good work. I don't need to tell you that because all the chapters have been published already, so they're already accepted by the scientific community. Of course, I also like the study because I'm kind of associated with it a bit because it's actually an extension of your master's thesis that we worked on together, where we tried to use these methods to parcellate the cerebral cortex, and where we, of course, tried to compare also similarity between parcellations with two methods, functional connectivity, but also structural imaging or diffusion tensor connectivity data. I also like very much, that's a kind of a big step from cerebral cortex to the periaqueductal gray, and it's a very different structure neurologically. The cortex has large fields, it has large columns, whereas the periaqueducal gray is really a very small area. I think somewhere you say that's 14 millimeters or something, it's very small. And it has a conglomerate of small nuclei, and that makes it really challenging for functional connectivity because every signal that you have is a mixture of the functional connections of these areas. So I think it's not a trivial step to go to this small brainstem areas. It's a really challenge. I think you did a very good job in making that step. So I have to ask you a few questions, of course. There's actually two questions that I would like to ask you. I'll start with the first one and then see how far we get with that. There's one thing that really intrigued me when I read your two chapters, and that is that you, when you pre-process the bold signal to use for functional connectivity, you high pass at 0.1 hertz. And that's, of course, there's nothing wrong with that, but I would like to know why you did that, what was behind the decision, because normally functional connectivity uses low frequency fluctuations, say from 0.1 to 0.01, so events that evolve in 10 seconds to 100 seconds, very slow changes in the signal. Whereas if you high pass at 0.1, that means that you only focus on events that take 10 seconds or faster to evolve, waves in the signal. And that's not trivial because, first of all, you want to show that your modules that you find or the clusters that you find, that they are independent of bladder, sensation of bladder fullness, which is a very acute phenomenon which might particularly influence higher frequency fluctuations. It's not as fast as cognition, of course, but it evolves at a kind of higher, the sensations evolve at a higher frequency phase. So you actually make it more difficult for you to confirm your hypothesis, I think. On the other hand, and that's the second point, is you do find that there is changing connectivity between modules depending on bladder fullness or the sensation, which is of course much more easy to detect when you look at higher frequencies. So I wonder whether these findings, how these findings relate to your decision to look at higher frequency signals.
Esteemed opponent, thank you very much for your compliments and for this very good question. To be fair, I think you make a very, very important point. And I think we might have to consider to investigate the effects of this filtering on the results. It is true that I do think that also the changing in bladder sensations is not as fast a process as you think. We are very much interested in continuous changes of bladder sensations. So these changes that we're currently looking at are also slow, or the scans, the data is long. It's several minutes long. So I think your point is well, very well taken, and we will definitely have to look into this further. I think we made this decision very early on, and it is methodological development. So it's work in progress.
Mm-hmm.
And we made the decision based on literature that we were working with and that was available. But we should indeed conduct other analysis to check what the effects of this filtering are.
Then my second question has to do with also with a bit more technical, because you focused on, well, it's more maybe a general question, because what kind of improvements are possible or do you see in the future? Because if you look at the pictures, there is you prove beyond doubt that there's replicability in modules. But if you look at the picture, there's also a lot of noise in the modules. So, and of course, we also want to push the limit and try to get more accurate or more replicable data. For instance, you work with the intrinsic functional connectivity. That means you only correlate signals from within the periaqueductal grade. Do you think that there's improvement possible when you would include extrinsic functional connectivity, taking the signals from everywhere or in particular parts of the brain, and you get more differentiation between modules because they are at larger scale more diversely connected. Or another approach might be that
can you please specify your question? Otherwise, we have to interrupt.
So my question is, would it be a different result or an improvement of the result when you would focus on extrinsic functional connectivity instead of intrinsic functional connectivity?
Yeah, that's a very, very relevant point. Very interesting. That's actually the point that we are at the moment, and we're starting to look at the interaction between different PAG clusters and the pons, and then look between. So right now, our latest analysis, we mapped the change in functional connectivity between empty and full bladder states with every voxel in the pons, with the different clusters in the PAG. And the interesting thing is that we see exactly opposite changes in functional connectivity between empty and full for different PAG clusters. So that kind of confirms to us at the moment that the functional subdivision that we have made of the PAG seems to be very meaningful because it holds up on analysis with different parts of the brain, so the pons, for example.
Yeah. So that would probably lay the basis for the idea of extrinsic functional connectivity.
Yeah, thank you very much. Yeah.
You think that there's something to gain?
Sorry, there's no time for questions anymore.
Yeah.
That's over now. Thank you. We have to continue, and we going to continue to the United States. I would like to welcome Dr. Clarkson. She's affiliated with the Department of Geriatrics at the University of Pittsburgh, and I'd like to also thank you for joining this morning or this afternoon. Thank you.
Hi, I thought that your attention to the signal by using the signal to the 1,000 random parcellations to ensure that you were actually getting a signal was really quite good. But I'm really quite interested in what we know in functional MRI, the test-retest repeatability is not particularly good. So I'm wondering, like, what do you know about the test-retestability of this particular parcellation technique? Have you tried it in people in 2 different sort of time points or in 2 different scans?
Esteemed opponent, thank you very much also for this question. To some extent, I think that how we show that there is a very significant overlap between empty and full bladder parcellations shows test-retest repeatability, because we have this is measured at 2 different time points. And I think also the overlap between different participants in functional clusters of the PAG also shows that it's not just noise that we're looking at. I think that also tells us something about the reliability of these parcellations.
So what is the comparison of the within subject overlap versus the between subject overlap? Can you speak to the sort of repeatability of the technique because of that, or do you think that's something that needs to be more looked into?
Yeah, so that's not something I can connect a number to directly, but I do think if you look at table 1 on page 70, we do see for, well, here we included data from 6 participants, and for 2 of these, we find significant matches for all 3 PAG clusters. For 3 of them, we find significant matches for 2 out of 3, and for 1, we find 1 out of 2 significant matches. And then if you look at chapter 6, you do see that there's, well, across subjects, there is less overlap in how similar these clusters are. And I do think it's, well, I think it's quite nice that we found overlap at all, significant overlap. At the group level, but it does show indeed that mapping the PAG at the group level is not somewhere we are yet. But we do know that at the within subject level, I do feel quite that these results are quite strong.
Yeah. Okay. So that leads me to my second question, which is more related to chapter 6. But it's also related to the anatomical overlap of the significant clusters. So you showed the correlation between, well, the overlap between participants 5 and 7. So I was wondering, what do you foresee this overlap being in sort of in a group structure, and have you any experience or knowledge of any other PAG parcellations and the actual, like, the anatomical overlap? And do you think the technique is maybe actually is too sensitive to sort of positioning to be able to appropriately look at the overlap between different participants?
I'm not completely sure if I understood your question correct, but we do we do see a significant overlap across participants. We tested that with a permutation test where we took all information out of the data that relates to spatial proximity and real functional connectivity. So we used correlation values between voxels that represent what we find in the normal data. We shuffled these matrices and we made random parcellations based on those correlation matrices. And when we then compare the correlation values or the dice coefficient overlap that we see between our real data with those from the permutation test, we see for many we see a higher correlation. But as you say from the figure that you referred to, there's also of course a large area that does not show this overlap. And that is kind of a work in progress to try to move towards really why is that the case? Like, so what is really the center of a functional unit within the PHE that we can determine with fMRI that really holds up at the group level? And I think, for example, in a larger population, something like heat mapping of these different clusters might help us to really try to determine what parts of these clusters really hold up on the group level and really move towards a group level map of the PHE. Does that answer your question?
It does. Thank you very much.
Thank you very much. Then the opposition will be continued by Professor Heesakkers. He is affiliated with the Department of Urology at our university, and also give him the opportunity to have some discussion with you.
Thank you very much. Congratulations with the achievement of the thesis, also the promotion team. I want to express my gratitude to it. And I have a question on chapter 4, and you started the analogy, so I continue with that. So let's say that we have the map of the Netherlands, and Limburg is the urethra, and the rest of the Netherlands is the bladder. Then we put a balloon into the Netherlands via Limburg, and then we fill it with gas, and then something happens to the continent state of the Netherlands. So how does it work exactly? Is this balloon in Utrecht absorbing a lot of pressure from around, or is this balloon coming up with gas in Groningen again? They don't see it coming. So what is really happening? Is the Netherlands or the bladder lifted by the balloon, or is this balloon really absorbing pressure? So what is the real mechanism, you think? I know you speculate already a bit on it, but perhaps we can continue that till the end of this promotion defense.
Yeah, I think there are a lot of different mechanisms that might be possible. It might be the buoyancy of the balloon that pushes the bladder wall upwards. That's one proposed hypothesis. It may be the continuous interaction between the balloon and the bladder wall that gives a constant stimulation to the bladder wall and increases some activity in the muscle. I think these are all possibilities, and I think what we contribute in chapter 4 is the development also of methodological approaches to analyze video urodynamic data, well, largely data driven, to try to get more information about this. And I cannot say exactly what the underlying mechanism is, but I do think we are able here to make methodological developments that can help us to answer a question like this.
Yeah. Well, I think it's a good addition to video urodynamic investigation, because we look at certain things, but never at balloons. So you did, that's good. Yeah. I saw in the figure that you have in your chapter that there's two differences. There's differences between the two pictures. Is that from the same patient?
Yeah.
Okay. And then you say, well, perhaps the whole bladder is lifted by the balloon. Does that mean that the balloon goes up to the ceiling of the bladder or the dome, as we call it, and just lifts the bladder, or so did you have indications for that or not?
The balloon does indeed go to the dome of the bladder, and this somehow increases or makes the bladder take a more vertically oriented shape. Yeah. But how I cannot tell. I cannot tell if it's from the buoyancy or if it's from stimulation to the bladder wall, change in muscular activity of the bladder. from the video, I cannot tell what is exactly causing this. I can just
Well, that's the good thing
determine
about
it's
it, because we don't know all
Yeah,
of
it's
us, so,
very good.
so we can just speculate. Huh?
yeah.
And in between, if you say, okay, it's bladder pressure absorption or lifting of the bladder,
You are allowed to finish your question, and you are allowed to finish your short answer.
which to your view would be the most important mechanism of the two?
I think a continuous stimulation to the bladder wall would be a very, very interesting mechanism that also could be of benefit to future therapeutic developments.
Thank you very much.
Thank you very much. Mr. Candidate, Mr. de Rijk, the time appointed for defending your thesis has now passed. The degree committee here provided will now withdraw to discuss the quality of your thesis and in particular the way you have defended your thesis this afternoon. I request that you and your company await the results of our deliberations and our return to the aula. Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Ja. Ik vond het wel vervelend dat wij geen scherm hadden.
Ja, dat is wel lastig. Ja.
Maar jij was wel vertellen, maar ik denk, het is zoveel fijner als je daar plaatjes bij hebt.
Oh, dat is wel snel.
Dear candidate, dear Mr. De Rijk, I will continue now in Dutch. Okay? So also iedereen duidelijk hier, uit onze familie ook, dat iedereen weet wat we nu de volgende stappen zijn. De commissie hier aanwezig heeft zich uitvoerig beraden over de kwaliteit van die proefschrift, maar vooral ook op de wijze waarop u dit proefschrift hedenmiddag heeft verdedigd. En op grond van haar unaniem positief oordeel, maar ook gelet op de examens die u vroeger heeft afgelegd, heeft ze besloten u het gevraagde doctoraat toe te kennen. De hoogleraar professor van Koeveringen is gemachtigd u op de gebruikelijke wijze met deze waardigheid te bekleden en ik geef daarom gaarne het woord aan uw promotor.
Belooft u dat u altijd volgens de beginselen van de wetenschappelijke integriteit te werk zult gaan, eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Ja, dat beloof ik.
Krachtens de bevoegdheid door ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hierbij u, Matthijs Maria De Rijk, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens de wet en gewoonte zijn verbonden. Ja, ten bewijze hiervan overhandig ik u nu de bul, door rector, secretaris en overige leden van de promotiecommissie ondertekend en met het grootzegel van de universiteit bevestigd.
Dank u wel.
Dokter De Rijk, van harte gefeliciteerd met dit prachtige resultaat, je dokterstitel. En ook mijn hartelijke felicitaties voor je vrouw, je zoontje en je familie en vrienden. En ik ben al trots op je, dus zij zullen dat zeker zijn. Deze promotie betekent veel voor je, maar die betekent ook veel voor mij en ook voor de afdeling urologie en voor de universiteit. I also would like to congratulate you on behalf of the whole promotion committee. And in addition, I would like to thank every one of them here for their valuable contribution. First, my fellow promoter, longtime collaborator and friend, Professor Laurie Burder. She played an important role in the translational diversity of your thesis, by which you were able to show your versatility, but also your resilience. Not to speak about the effort you had to make to get to her lab in Pittsburgh in between full corona lockdown periods. And Thijs, you were able to consolidate our collaboration there. And I'm sure that we will conquer collaborative grants together now and in the near future. Dr. Sajjad Ranamai, your motivation and perseverance made us win the Astellas Europe Fund grant for the second time on a proposal to explore the bladder-brain connection by high-flux functional MRI imaging. And that research line is flourishing now. and gave us the opportunity to hire Thijs. And Dr. Job van der Hurk, your innovative analytic skills in combination with an outstanding talent to explain difficult processes made you a highly valued collaborator. And I'm proud that you're also a team member now for the future. Now I switch back to Dutch, because I address you and the audience as well. Er zijn promoties die duidelijk een doel in zichzelf zijn en het eind zijn van een vaak moeilijk en bochtig traject. Hierbij wordt dan later al dan niet de weg tot het doen van onderzoek weer teruggevonden en helaas maar af en toe bezield verder geëxploreerd naar verrassende, innovatieve hoogtepunten. En gelukkig zijn er hiervan wel een paar prachtige voorbeelden in de zaal. Bij Thijs is dat anders. Een aantal jaren geleden was ik op zoek naar een promovendus om samen met Dr. Ranamai het FMRI-project te gaan voortzetten. Er was iemand nodig met een exploratieve geest, maar vooral ook voldoende capaciteit om zelfstandig te kunnen werken. Doordat ik op dat moment afdelingshoofd was en Sajjad Ranamai in Aken een klinische aanstelling had als uroloog. Op dat moment was Thijs net klaar met zijn master neuropsychologie. En hij kluste bij op de Grant Office. En Marco Berndes heeft hem daar gespot als een researcher in hart en nieren. En met mij in contact gebracht. En daar ben ik hem nog steeds heel dankbaar voor. Thijs maakte in het begin een wat schuchtere en iets te serieuze indruk. Maar al snel bleek hij best gezellig om een biertje mee te drinken. En ook al heel vroeg bleek hij een eigen ideeën te hebben en analyses te ontwikkelen, die hij op zeer respectvolle, maar ook meestal wel vasthoudende manier naar voren bracht. En dit gaf aanleiding tot zeer nuttige researchdiscussies, die mij vaak ook nieuwe inzichten gaven en niet zelden wat filosofisch van aard werden in de lijn met wetenschapsfilosoof Karl Popper. Vandaar mijn cadeau vandaag. En deze discussies hebben niet alleen gezorgd voor dit mooie veelzijdige proefschrift, maar ook een fundament geslagen voor de toekomst. De promotieonderzoeksperiode zat bij Thijs natuurlijk ook vol uitdagingen. Er heeft zich al eerder aangehaald door corona een flinke vertraging voorgedaan. En in plaats van bij de pakken neerzitten, hebben Thijs en ik deze periode gebruikt om een onderzoeksvoorstel voor NWO te schrijven, wat uiteindelijk ook is gehonoreerd. Thijs heeft hier een essentiële rol in gespeeld, die veel meer is dan wat je van een promovendus mag verwachten. Het NWO Humane Meetmodellenproject is inmiddels in volle gang en momenteel komen er door inspanningen van Thijs en Job van der Hurk belangwekkende resultaten uit naar voren. En van zeer innovatieve analyses die niet alleen voor de urineblaas- en hersencommunicatie van belang zijn, maar ook van belang kunnen zijn voor communicatie tussen de hersenen en de rest van het lichaam. En ik ben daarnaast ook zeer blij dat we dit project samen met een gerenommeerde Nederlandse expert op dit gebied doen, professor Bertil Blok. Of eigenlijk nog geen professor, maar hij gaat dat wel worden. Ik weet niet of ik dat al mag zeggen. Maar van het Erasmus MC in Rotterdam. Ik ben blij dat we dat samen kunnen doen. En ik wil ook pleiten voor een respectvolle concurrentie en productieve samenwerking, om dat met elkaar te laten afwisselen in onze research community. Your foreign research experience, through your working visit in the lab of Professor Burder in Pittsburgh, gave your PhD period an important additional value. And I have stimulated this from the beginning, as I have experienced benefit from my own research fellowship in the US during my whole career. Dit alles, naast je mooie verdediging vandaag, zal voor jou een opmaat zijn naar een mooie researchcarrière. En ik roep dan ook onze urologie vakgroep, onze vakgroepvoorzitter, professor John Heesakkers, en het hoofd van Mental Health and Neuroscience, professor David Linden, op om zich samen met mij in te spannen om dit binnen onze school- en researchgroep mogelijk te maken. Zo kunnen we jouw promotie geen doel, maar een middel maken om te komen tot een mooie researchcarrière voor jou, toegevoegde waarde voor functionele urologische research en nieuwe therapiekansen voor onze urologische patiëntenpopulatie. Dr. de Rijk, beste Thijs, nogmaals van harte gefeliciteerd. Ik wens je enorm veel succes in je toekomstige researchcarrière. waarvan ik hoop dat we daarin nog een ruime periode samen kunnen optrekken.
Dank je wel.
Ik heb gezegd. Now I give the word back to the pro-rector.
Okay, thank you very much. And I would like to continue too. Also on behalf of Maastricht University, I would like to congratulate the young doctor. And it looks on your body language that you also like to see that sounds that I congratulate you as a young doctor. In my congratulation, I would like to mention two things. First of all, I noticed in your curriculum vitae that you really worked over the years on water management, because you started in the beginning to use water management, and now you're still working on urology, let's say also controlling internal water management.
Yeah.
That's one important thing. And secondly, I really would like to congratulate you that you are, as we call it here at our university, a three steps rocket. You didn't went straight to high school, from high school to university. No, you had several small steps before finally entering the university here. You went from VBMO to HAVO to basically to VWO. That's what at least I read here at your thesis.
Yeah.
And I have to compliment you. There's also one thing I would like to mention as being a neuroanatomist. I don't agree with you that the PEG is Utrecht. Absolutely not. No. We have a Utrecht, and we call Utrecht the thalamus. And basically what you are the A2. The A2 leads to the thalamus, and that's the correct formulation. But anyway, it's my opinion. I would like to congratulate you. I'd like to congratulate the promotion team. I think it's very good that you have 4 members in your team who guided you over the different aspects of your career. En nou, I also switch to Dutch again. En ik wil ook graag nogmaals bedanken de leden van de corona dat ze hier aanwezig waren, dat ze de tijd hebben genomen. Also the members, I'd like to thank the members also in the UK and in the US that they joined today. Ik wil graag feliciteren de familie die hier aanwezig is natuurlijk. Het is een prachtig voorbeeld van hoe u kunt zien hoe u vanuit het Heerlense toch in het Maastrichtse terecht bent gekomen, laat ik het zo formuleren. En dat uw familie, uw zoon, man, het uitstekend gedaan heeft. En ik wil ook graag kijken, en ik switch again to English, to all the PhD students who are sitting here in the audience. Basically, that's a good example, and I hope to see you back in the not too far distance and also defending your thesis here. And by saying this, I'm going to conclude this thesis. I will use my hammer, which you haven't seen, but I still have him here, as you can see. And I close this session. How do we proceed? We proceed as follows. De receptie gaan en daar genieten van een kop koffie, natuurlijk een biertje, een glasje wijn en misschien een stukje vlaai. Wij blijven nog even hier, want we gaan een foto maken hier met de leden van de oppositie. We maken een foto hier met de online members en we maken nog een foto op de trappen. En daar